My name is Dr. Gwynne Mhuireach. I'm currently a postdoc at the University of Oregon where I study microorganisms living in our buildings, backyards, and neighborhoods. Today I'm excited to share some preliminary results from my USDA-funded postdoctoral research, the Gardener Microbiome Project. This project is a community science effort to understand whether the microbes we encounter in our backyard gardens can transfer onto our skin during gardening activities, and how long they can persist there. It is well known that microbes help to create healthy soil. When I talk about microbes, I mean bacteria, fungi, archaea, and viruses, among other microscopic organisms. For this project, I focused primarily on bacteria. All of these different microbes work together, along with other soil organisms, to perform many important processes, such as decomposing organic matter, degrading pesticides and pollutants, cycling minerals and nutrients, forming soil structure, promoting plant growth, inhibiting pathogens, and improving water availability and uptake for plants. We also know that management practices can affect the diversity, composition, and activity of soil microbes. For example, organic systems often have higher levels of soil organic carbon and greater microbial activity and biomass. Application of manure especially has been demonstrated to have a significant positive effect on soil microbial biomass and activity. Tillage is another factor that can have a huge effect on soil microbes. Despite our increasing knowledge about soil health, we still know very little about how exposure to soil microbes might affect the health of farmers, gardeners, and other individuals who spend a great deal of time in direct contact with soil. Before I go on, I want to emphasize that we are always being exposed to microbes, and that's a good thing. In mouse studies where researchers have eliminated all microbes from the bodies and environments of the mice, they do not develop correctly, they become very sickly, and they die much earlier than their microbially exposed counterparts. In your day-to-day life, you can expect to inhale somewhere between 100,000 to almost 3 trillion bacterial cells each day. You ingest a similar number with your food and another few million in your drinking water. However, our levels of exposure can vary widely, depending on our environments and activities. The types of microbes to which we are exposed can also be quite different. For example, riding a crowded bus and weeding your garden will both expose you to a high abundance and diversity of microbes. But riding a bus, you'll probably encounter mostly human-associated microbes, whereas in your garden, you're more likely to meet soil and plant-associated microbes. For most of our history as human beings, we spent the majority of our time outdoors and surrounded by natural environments. Now we have drastically changed our living environments from natural and agricultural lands to urban and suburban land types, and we now spend almost 95% of our lives inside buildings and cars. In the US, we've also gone from a population of about 90% farmers to now less than 2%. At the same time, autoimmune disorders such as allergies and asthma have become increasingly prevalent. Some researchers suggest that reduced exposure to microbes from the natural environment may be part of the explanation, since we depend on these exposures in early childhood to help our immune systems develop properly. And finally, a newer theory called the Biodiversity Hypothesis states that microbial diversity is linked to macrobial diversity, or the diversity of organisms we can see, like plants and animals. This idea makes a lot of sense because every garden plant and every insect has its own individual microbiome, just like humans do. So if there are a lot of different plants in your garden, you are likely to have more diverse microbes than your neighbor with a monoculture of turf grass. The goals for this project were to measure how many and what types of soil microbes are transferred to skin during gardening, as well as how long they can persist there. I also wanted to explore how management practices, crop type, and climatic region impact garden soil health and microbial communities. And lastly, I wanted to pilot a community science pipeline for collecting, analyzing, and sharing garden soil health data. To accomplish these goals, I recruited 40 master gardeners from 2 different climate zones in Oregon to collect soil and skin microbial samples. They collected skin swabs before and after gardening, as well as 12 and 24 hours later. They also collected garden soil from 3 different beds in their gardens right after their first skin swab. Then I used high-throughput sequencing of the bacterial 16S rRNA gene to explore similarities and differences between the samples. My analysis demonstrated that soil microbial communities are clearly different than skin microbial communities. In this ordination plot, the garden soil samples, shown in green, are clustered tightly together on the left of the plot, indicating that soil microbial samples tend to be more similar in composition to other soil samples, and less similar to skin microbial samples, which are represented by the orange, red, purple, and black circles spread across the right side of the plot, each of which indicates a different collection time. You may also note that some of the purple points, which represent skin samples collected immediately after gardening, are shifted toward the green garden soil points, suggesting that those skin microbial samples were more similar to soil samples than to skin samples taken either before or 12 or 24 hours later. And here we can see more clearly how the number of bacterial taxa shared between an individual's skin and their garden soil changes over time, with some people seeing a dramatic increase immediately after gardening, but a rapid return to baseline within 12 hours. In this study, we did not give the volunteers any special instructions regarding hand washing or daily activities, so these results represent a real-world type of effect. We also note that because this study took place last summer during the COVID-19 pandemic, people may have been washing their hands and using hand sanitizer more often than normal, which may have also affected the results. On this slide, we are looking at the results for community scientist number 6. The Venn diagrams on the left show shared taxa between garden soil and skin samples over time, while the bar plot shows taxonomic composition at the genus level, with the top 25 most abundant taxa shown as colored bars and all the less abundant taxa colored as gray. At the individual level, we see that skin bacterial composition changed dramatically immediately after soil contact, but the effect was transient, indicating that the skin microbiome is resilient against incoming soil microbes. However, if you are someone who experiences daily direct contact with soil, then you likely have a regular influx of soil microbes, which may still have potential to interact with your skin commensal microbiota and your immune system. Lastly, I found that management practices and location both had significant but weak effects on the soil microbiome composition. In this ordination, we're only looking at the soil samples, where the location is indicated by hollow circles for the high desert and filled triangles for the Willamette Valley, and organic versus non-organic practices are indicated by color. There's also an interesting interaction here, where you can see from the plot that much of the effect is driven by the difference in organic versus non-organic practices only in the Willamette Valley. In closing, I would like to thank everyone who has helped make this project happen, including members of the Garden Ecology Lab at Oregon State University and members of the Institute for Health in the Built Environment at the University of Oregon, particularly my mentors Gail Langalato, Kevin Vanden Wy-Mellenberg, and Krista McGuire. Special thanks to all the community scientists who participated in the project and to my advisory committee. I would also like to thank the USDA and Alfred P. Sloan Foundation for generously funding this project. If you would like to see project updates, just scan the QR code on the right.